That should be visible. All right, so again, welcome. I wish we were all in person here. I live in Park Merced, so I'm not far. I get to walk around campus every day, and it's just not the same. It's not the same without students around. But anyway, if you want updates on things going on at the university, if you haven't seen this, this is the COVID updates website for the campus plan. We are online all fall semester, as you know. If you need help with basic needs, food, shelter, and stuff like that, the university is there to help as well. And then grad student resources, everybody should be familiar with this website. Make sure to go there for current students because it's got all the dates, all the forms, all the stuff that I hate to do, like the paperwork kind of stuff that we have to do. So make sure to keep an eye on that as it changes and gets updated. And then finally, probably what everybody's wondering about is what's the deal with research? So research on campus is slowly restarting. I'm on the biosafety committee. I'm on the University Research Council. I'm on a bunch of committees and I have zero power to say anything. Basically, we give recommendations and stuff. But right now we're at the point where people like me, faculty in our department, are submitting applications to be able to get back on campus. And the applications entail a whole plan of how we're going to deal with COVID if somebody gets sick, a whole plan of what kind of PPE we're going to have to wear and all that. And I can just tell you for now that there's going to be no human research for the fall so far. And the spring is not looking good for at least working with participants. We might be able to get back in the labs, sooner rather than later, but we're probably not going to bring somebody in to do like a VO2 max test or something for a while. So just to kind of think, I'm gonna be throwing out ideas for research and stuff. But if you're thinking that your thesis is going to start this fall and you're going to be doing like 1 RMs in the gym and max tests and stuff, like we're not doing any of that. So it's a bummer. I'm really bummed because I haven't been in the lab since March. Really, I've been in to check on stuff and we've had a couple safety meetings, but other than that, we're going to have to figure out how to do this remotely. So just keep that in mind, and I'll keep you updated every week. As I hear something, you'll hear it from me. So, and if you do have any questions, we can always chat on email and stuff. Okay, so as you know, this is going to be recorded. I'll just, from the time I started this, I'll basically crop that and save that onto iLearn so you can rewatch this video if you want. And then Zoom courtesy, again, stay muted unless you're asking a question. Right now, it's hard for me to use the chat function as well as the every other program I've got open. So if you do have a question, just go ahead and speak up and we'll hear you, or you can raise your hand, but I can't really see everybody while I'm going through this. And then again, turn your camera on for the first few minutes as we did when we talked. If you want to keep your camera on now, that's totally fine. I'm going to keep mine on most of the time unless my internet gets kind of crappy, which it probably will at some point. I'll have to kick that off. So okay, so here's the agenda. Every week we'll have a little agenda. We're going to review the syllabus. The schedule is to be determined, but just plan on meeting on most Wednesdays at this time, and I'll get a more detailed schedule as we get through here. I'm going to talk about my research. If you've had me in undergrad classes like 355, 555, or 482, then you've probably seen some of this, but most of my research ties in with muscle physiology, so that's cool. This is the first time this semester we're going to do something called a class topic or a class theme for the fall. And our class theme is going to be muscle memory hypothesis. So I'm going to introduce this theme today. This is a really so I'm planning on doing is every semester have like a hot topic of research and we're going to get really deep into it. So this is kind of, win-win for me and for you because this is something I haven't looked into recently and it's probably something you haven't either. We're both or all of us are going to become experts on this topic. Then we're going to do some team breakout rooms. I'm going to create teams of 2 or 3 at the end of class today, and we're going to work on our first assignment. Is going to be doing a short presentation 2 weeks from now on some biochemistry review. So the plan for next week is going to be, I'm going to talk a little bit more about biochemistry. We'll discuss another project for the semester, which is a lit review. So I'm going to have, you're basically all going to choose a topic. We'll get into this later of your choosing and do a literature review that's going to be due at the end of the semester. We have a ton of guest lecturers. I think like 8 or 9 interested and 5 for sure so far. So these are, I'm actually really excited about. We're going to have our first guest lecturer next week, Donnie Gregg. He was a master's student like 2 years ago here, and he works for a company, Aperion, that Megan knows about. She did some internship work there as well, so probably be able to talk. But I'm trying to get people from industry, government, academia to come in and talk about what they do related to muscle physiology. And then this is the link, this link at the bottom of this page, I'm going to put that up on iLearn as well. That'll be our link we'll use for the remainder of the semester. So it's not the one we use today. I created a different one. So this will be, we'll use for the rest of the semester unless we get Zoom bombers, and we'll have to switch. I don't know if anybody's seen any funny videos of Zoom bombers, but it hasn't happened yet, knock on wood. All right, so any questions on agenda? Cool. If you got anything, you can go ahead and speak up, but if not, I'll move on. So every week, I'm also going to share opportunities with you because stuff comes across my desk and it would be very easy not to read it and just push it away. But whenever I see something interesting or a colleague or friend bring something up, like a job opportunity, I will let you all know. This one for this week, this just came out yesterday or the day before, I believe. So one of my friends from undergrad, actually, so we worked together on a senior project in undergrad, like 12, 13 years ago or whatever it was. And now he's basically one of the head salespeople at this company called Echo. And they make really cool electronic stethoscopes. I actually have one. I should have had it here ready for a demo for you all, but I don't have it. But it's an electronic stethoscope that's actually really cool that can show a one lead ECG on it, like on your phone. So doctors can actually like send this to patients and they could do a little echo on them or a little ECG on themselves. So I think it's pretty cool tech that it runs like $100 or $300 or something like that for this. But anyway, they have a job opening that you are all qualified for if you have your bachelor's degree already, an internship in medical affairs. I don't know anything about pay or anything like that, but if you do want to apply, the application button is there on the bottom. So that's the opportunity of the week. Also, we've got an upcoming virtual conference. Conferences are not going to happen in person for a while. I think it's just too big. And with COVID, people are too scared to get obviously thousands of people in a place. And I was supposed to give a lecture in November in Spain. I was really excited to go to Sevilla and hang out. But anyway, this will be virtual. And the conference is a cool acronym, TAPAS, but it's the International Technology and Physical Activity and Sport Conference at the University of Sevilla. It's like 11 euros to go online. So it's like $10 or $12. I don't know which way it's going, but it's not that expensive. And it's a 4-day program. I'm going to be giving a lecture on virtual reality, health, and exercise. So if you are interested in that topic, I would definitely check this out. And then there's going to be extra credit opportunity for this that I'll post up. In grad class, you should be working hard enough where you don't need extra credit. Everybody should be shooting for A's in this class or B pluses at the minimum, but this will be an opportunity to make up some points if you need to. So keep that date. It's actually over Thanksgiving, because they don't have Thanksgiving in Spain, I guess. But we do have a couple other students that are going to present there as well. So that's another opportunity for this semester. All right, so let's do a quick review of the syllabus. So again, the schedule is to be determined, but just plan on meeting every Wednesday. That's going to be kind of our time. Course description has changed. So again, this is just grad class now. This is not an undergrad anymore. It used to be paired with 681. What has been added, you might notice, it's basic and applied physiological properties of skeletal muscle from a molecular to whole muscle level. So we're kind of bringing this idea in the context of physical adaptations from disuse, injury, and training. And we're going to get into a whole lot more other things too. Course materials, no textbook required. So we're all going to go off of stuff off of iLearn as well as things that you'll find from the library. So we'll do a little tutorial next week on how to access the library database if you don't know how to use it. There's like 20 ways to get articles if you need a scientific article, and I'll show you the way I usually do it through the library. Because if we don't have access to it, you're all paying tuition. You have access to everything on the planet. So you'll get an interlibrary loan. It might take a couple hours or a couple days, but you can get access to anything ever published in the history of the world. That's why you pay tuition. It's pretty crazy. But my recommended text actually is this one by Dr. Richard Lieber, and it's out of print, so I don't use it for this class anymore the whole way through. We will go through the first 2 chapters, and I'll give you those in PDF form. So, but if you do, specifically if you want to become a PT or an OT, and you're interested more in the clinical route of muscle, this is a really good book. Dr. Lieber is a PT, and he was the head of the PT department at UC San Diego and University of Chicago, I think, for like a lot of years. But anyway, this is recommended. It is 10 years old, so it's, a little out of date, but muscle anatomy hasn't changed in thousands of years as far as I know. So a lot of the anatomy and stuff is still obviously relevant. So our theme, again, this semester is going to be muscle memory. And these are the course topics that we'll go through all these, and we'll go kind of in this order. So this week, we're starting to review biochemistry to make sure we're all on the same page. I did my undergrad in exercise physiology, or basically in kinesiology, and then my master's in kinesiology as well. By the time I was in your shoes, I had never had a biochemistry class, I think. Maybe. Maybe I had organic chemistry, but not biochemistry. I didn't even have that till I went to my PhD, and then I had a year of biochemistry. So that was insane. But we're going to review or do an overview of biochem just to make sure we're all on the same page. And then we'll get into anatomy, physiology, talking about fatigue, adaptations, injury, and disease. And as we get through the class, you'll kind of see some of these themes that I'm going to touch on in the next few slides as well. All right, grade, super important. Total of 600 points. We've got 3 quizzes and 3 exams. All these will be on iLearn. They'll all be timed, but I'm going to give you a fair amount of time. It's not like a minute a question. You'll have a fair amount of time, but it's going to be open book, open note, so it will be timed. And then the rest of the points are going to be this literature review we'll introduce next week, a presentation that we will introduce next week. Your participation is based on this. Everybody that's on today get participation points. And then team assignments. Throughout this semester, we're going to have 3 or 4 small assignments that will add up to points too. And then my contact info is here. I'm not in the office or the lab anymore, which sucks. I'm in the corner of my 1-bedroom apartment in San Francisco, like most other people are. But if you need to get a hold of me, probably the best way is email or schedule a meeting with me on my office hours, Tuesday, Wednesday, or Thursday. If you can't make those, but you still want to chat, grad students, I'll definitely try to make time for you all. That's kind of the point of grad school, is to get in contact with your faculty. Undergrads, when I've got 150, I'm probably not going to meet with them outside office hours. But if you all want to meet, like let's say you got work in the morning and you can't meet till, right before this class or after, I can always jump on and meet with you all. And then in the actual syllabus online, you'll see the next few pages are really important dates and stuff related to withdrawals and adds and drops. So make sure to keep an eye on those. Okay, any questions before we jump into the next section? Okay, I'll give you a second. We are going to take a break in a little while. So just to let you know, about every hour or so, I don't know about you, but I need to take a break. If you're sitting, we all need to get up and stand. And if you need to go to the bathroom or something, please don't keep your camera on and keep everything. You can, do what you need to do. I'll obviously post all these later, so don't feel bad if you have to get up or something in the middle. Okay, so this is the way I view muscle physiology. My comprehensive view of everything is looking at it from the whole body. So things that you can do, like VO2 max testing. This would be an example of a whole body measure, down to the muscle level, or muscle groups. So let's say your quadriceps, right? Your knee extensors and hip flexors. Maybe you're talking about one quadricep, like the vastus lateralis, that's one muscle. Sometimes we talk about them in groups. And when we get down to the muscle cell or the muscle fiber, I'm going to use those words interchangeably. So just know when I'm saying a cell or a fiber, it's the same thing. We have all the organelles and all those components, actin and myosin, that pull. Remember the sliding filament theory from undergrad, all that stuff. And then down to the molecular level, things like DNA, RNA, proteins, enzymes, transporters, hormones, anything like that you can think of at the molecular level, all that interacts with everything upstream from it. So, you might think about the whole muscle level. We might measure something like muscle strength or muscle size. This is a picture of a cross-section of an MRI. You can see pretty clearly the vastus is, the biggest, easiest muscle to get to. You've got all the hamstrings here, and then you've got your femur there. And then once we get down to the single cell level, this is a sample of my muscle fibers. So most of the pictures I share too in this class are from myself, because I've gotten like 30 muscle biopsies done when I was in my PhD school. So my legs are still good. I'm actually standing now. So, but this is an example of one muscle biopsy in this bundle. This is zoomed in like 20x what your normal eye can see. There's probably about 500 fibers in there, and then each individual cell is shown there. And we can do things like attach those to a force transducer and a motor arm and look at things like the cell function, which is pretty cool. And then again, inside the cell, obviously there's enzymes and other proteins and stuff that we have to worry about at the molecular level. All of it works together hand in hand. And a key concept here is that you probably remember this from Bio 101, structure equals function. And I didn't get this until like my third physiology class. I was like, oh, now I get it. But if you think about it, a muscle cell, most of the cell is made up of actin and myosin contractile proteins. It's long, cylindrical. Its whole job is to pull side to side. That's why a muscle cell looks like that. So the muscle structure, its anatomy or its microanatomy, dictates its function. Okay, any questions or anything before we jump into key concepts? Cool. All right, so key concepts in muscle physiology. These are, you'll notice throughout this class too, I break up things into fives because I'm going to make 5 groups. So there'll be groups of 2 or 3 in the class anyway. So I'm going to have 5 kind of concepts on here that I want you all to think about that we'll get into as the semester goes on. But one of them I just mentioned, that structure equals function. So that's the anatomy is going to dictate your physiology. Number 2 would be the levels of organization that I also just mentioned. So think about micro to macro anatomy. Think about things like cell theory, like all this stuff that we know and we take for granted. We're going to kind of talk about maybe when it was invented and why it, how big of an idea, just the idea that you're comprised of individual cells in your body, like muscle cells, was a pretty radical idea. Talk about communication a lot in biochemistry, DNA to RNA to protein. So that's also known as a central dogma of molecular biology. It's really like a central theory. It's the idea of how you transfer information from its stored form in DNA all the way up to the proteins that your body's made of. Hormones, also things like microRNAs. There's other little signaling molecules that we're going to get into and talk about that you may not have heard of. There's all kinds of messengers, receptors, transporters. Your body has to communicate with itself, with the outside world and everything. So we're going to talk a lot about that. Homeostasis, super important. Obviously during exercise, the whole point of exercise is to get out of homeostasis. Recovery, you're kind of getting back in, but we'll talk about feedback loops, regulation. And then the flow of energy too, or the flow of energy gradients like potential energy, diffusion, osmosis, this stuff will come up too. So if a lot of these words are familiar from your science classes, that's good. If you don't remember what everything is or you haven't heard of it, don't worry, we're going to get into all this stuff as the semester goes on. All right, so my research background, this is when everybody likes to talk about what they do, but the cool thing is this stuff is actually related to what we're going to talk about in class. That's why I teach muscle physiology, I guess. So my journey, I started out in community college at Cuesta College down in San Luis Obispo on the Central Coast. So like many of you, I didn't know what I wanted to do when I got into my community college, and I kind of fumbled around a little bit. I was actually going to be an anthropology major. I was really into archaeology. Like I went on some dig sites, these Native American dig sites, and I was super interested. But then I spent a couple days out there in the mud and the weather turned, and I was like, I'm like a fair weather archaeologist. I'll go out when it's like nice Central Coast, but when it starts to rain, like I don't want to be out there. So anyway, I got out of that, got into physiology. I took a physiology class at Cuesta College, and then I ended up getting into Cal Poly San Luis Obispo in their Kines program. And I really loved that. And then I didn't know what I wanted to do when I graduated from there because the last recession hit. So you all get the benefit of this recession hitting now. I had the last one, and I get this one too, I guess, back to back. But I didn't know what I wanted to do. So I ended up at Cal State Fullerton getting my master's. And that was probably the best decision of my life because it led me down the next path through research and stuff. So I didn't know what I wanted to do till I was in most of your shoes, like exactly right now, like 1 or 2 semesters into my master's, I started to kind of get it, figure out what I wanted to do. So let's say like summer in between my first and second year of master's, I did a road trip all across the country. Went to New Mexico, Texas, Ohio, Indiana, looped all the way back around. And I ended up applying to a bunch of places. I ended up going to get a PhD at Ball State University that's affiliated with the Indiana University School of Medicine in Muncie, Indiana. If you've never heard of it, don't worry. It's a small podunk town in the middle of Indiana, northeast Indiana, literally like called the Rust Belt because it had 150,000 population and then all the factories shut down and now it's got like 45,000 people. It's just basically a college town. But anyway, I might bring up Muncie every once in a while because we called it Funcy Muncie. It was just, imagine a Midwest college town, not much to do. Great place to study muscle physiology though. If you Google the Ball State Human Performance Lab or HPL, that's the lab that I came out of. And a lot of people in the 60s and 70s and 80s that started muscle physiology in the US went through Ball State. So it's got kind of like a very incestuous. Everybody knows everybody when you get up into like a certain field. Any field's probably like that. But anyway, I graduated with my PhD from there. And then I was applying for jobs mostly on the West Coast because I was done with the Midwest winters, and got funding from the NIH, the Army, NASA, and Cal State while I was there and at Fullerton, and then ended up making my way back to San Francisco State, which I'm happy to be here. I've been here 6 years, so it's gone by pretty fast. But anyway, the research I did at Ball State basically has led into the research I do now, and that's what I'm going to go over the next few slides. So the first thing I was introduced to when I got to Ball State was, this is it. As soon as you get there, they're like, all right, do you want a muscle biopsy? And obviously they can't make you do it, but if you don't do it, you're like ostracized. Like, no, like, I can't believe you didn't do it. So everybody gets a muscle biopsy pretty much when they go there. I was terrified. I had something in my mind that it was going to be like a giant thing that they would just jam into your leg and like work it around and pull out. But it's not like that. Basically, if I could explain a muscle biopsy to you, is you see this needle. It's from a textbook. This is actually Dave Kostal, Dr. Dave Kostal. We always called him Doc, but he was one of the guys that brought muscle biopsies back from Scandinavia. This is called a Bergstrom needle. It's pretty thick gauge needle, and inside there you can see there's a little opening, basically an opening, and then another round kind of circle inside that's basically a guillotine, that when you stick this into a leg, your leg's numb, so don't worry about that. You've got lidocaine, which is like Novocaine in your leg. This goes in for about half a second to a second. A little window opens and closes, snips out a piece of muscle, and you don't feel it until the Novocaine wears off. And then it just feels like you got a bump on your leg. It feels like you got like a really bad, either a Charlie horse or you hit your leg super hard on the corner of a table. That's kind of like what I can say, but it's, a split second, and then it doesn't feel like much. But anyway, I did a bunch of biopsies, and the important thing about biopsies is, can imagine in physiology, especially muscles, it's easy to get muscles from animals. We do animal physiology with mice and rats, which has been done for a long time. But getting stuff from humans is the key, because we're trying to either make humans better, stronger, faster, or help people recover from diseases and disabilities. What we could do with one muscle sample is get, like I said, thousands of muscle fibers probably from each one, and this has made huge strides in exercise biochemistry and metabolism, the muscle mechanics and microanatomy that we're going to talk a lot about, and then molecular exercise physiology, which is, again, what kind of signals happen to get muscles to grow or to shrink or to get stronger or bigger. So this was actually the first paper of the modern muscle biopsy. It was from Bergstrom in 1962. This was introduced, and Jonas Bergstrom was from Scandinavia. And the first biopsies to happen in the United States were in the early 1970s from Dr. Dave Kostal, who I mentioned. He was one of our advisors at Ball State, and Phil Golnick, who was at Washington State. And then from there, now there's probably 100, maybe more than 100 programs that do muscle biopsies for exercise physiology research. So it's really grown, obviously, since the 60s and 70s. This happened because these guys went on sabbatical at the Karolinska Institute, which, if you have never heard of that, look it up. That's where the Nobel Prize in Physiology and Medicine comes from. Karolinska is in Stockholm. They brought the technique back to the United States. And since then, our colleagues and collaborators just in the last 5 years have done over probably 600 biopsies now, since from 2015 till March, which was, the last time we did one because of COVID. This is just kind of a cool story. This is Dr. Kevin Murak, one of my colleagues that I'll bring up a lot, especially as we get into that muscle memory stuff. But we went down to Singapore to Nanyang Technological University. This is Dr. Yifan Yang. She was a Ball State graduate from the 1990s, and we helped introduce the muscle biopsy technique to their university. So we brought this all around the world, literally. I flew to Singapore, and we had to go the wrong way around because there was a storm in the Pacific. So we ended up flying from Indiana to Paris to Singapore, hung out there for like a week, and then flew from Singapore to Japan to Indiana. So we literally did the full circle just to bring the biopsy technique there. Okay, so like I said, muscle biopsies contain thousands of muscle cells or fibers. With those, here's what a small sample looks like. This is in our lab. And again, we can get single muscle fibers. Here's a muscle fiber zoomed in from our microscope in the lab at 400 times what your eye can see. And you can see these lines, those are the striations, the Z discs. So remember when we were learning about the microanatomy of muscle with the Z disc, H band, I band, and all that, that's how you can actually see those. And then we can label them with different labels to mark things like muscle nuclei. So these blue dots are myonuclei. And you have thousands of nuclei. We're going to get into that. This is the whole premise of that muscle memory theory is related to these nuclei. So in 2015, when I got to SF State, I got a sweet job here. You know, I'm working with Dr. Lee, who does the clinical stuff you're probably all working with as well if you've taken his classes. Dr. Kern, who just retired. If you missed her, bummer, because she was all into metabolism. She was super legit. And we're still working with her on some research projects. So you might see Dr. Kern around every once in a while too. But in the lab that I started, basically our goals are to teach and mentor, which teaching right now, so check that off the list. Community engagement, we do a lot of lectures and I post a lot of things on YouTube and Instagram and stuff. And then scientific research, obviously that's kind of like the basis of everything. The lab has kind of 3 overlapping areas that we study. One would be methodological advancements, hard to say, but basically we try to advance methods to make things better and easier to do. Muscle cell biology, which is pretty much everything we're going to talk about, and then applied human performance. All right, I've got a question for everybody real quick. So I want to talk about this idea. I'm going to stop sharing real quick all right, I'm going to gallery view. So there's 2 types of research you can do. And I don't know if you guys know or can explain these, but there's applied research and basic research. Does anybody want to take a stab at what those 2 are and how they differ? Yeah, go ahead, Julissa. You can just speak up if you want to.
So I'm assuming applied research is you're looking at one specific thing. So like if it was myonuclei, you'd be just focusing on that, right? Compared to a more general one is what I would guess.
Kind of.
No?
Anybody
Okay.
else? Yeah, I think you're on the right track, and maybe it's just the wording's different. But anybody else? Matt, do you got something?
It might be a little too simplistic, but I believe basic is highly theoretical and highly controlled. When applied is a little bit more practical, but you can't control the variables as easily.
Okay, I think that's getting closer. I like that. I mean, I think they're both building. Anybody else have any more to add? Yeah, Kristen? I'm like pointing. I don't know who I'm pointing at. Yeah. I don't know if I can hear you still. Let's see. Can you unmute? Oh, let's see. Is it going off? Weird. Okay. No problem. All right. I'll give you all, anybody else? Okay. So I think you guys were kind of both on the right track there, but yeah, think about applied research as something that your question is, well, I can't say the word applied in the definition, but that there is some kind of application where you're getting something out of it. Like if you want to know if a drug works, like I want to know if this COVID vaccination works before I take that. So I'm going to take the vaccine that was developed and I'm going to give this to 1,000 people and see how well it works. That's totally applied research. But to make the vaccine, you need basic research. And basic research is more related to, again, the theories of how this might work. And as we go through, all the studies that you read, maybe make a mental note. Was this study basic or applied? And try to figure out what kind of research you're reading more. In this class, you're going to get both. So if the studies, says the purpose of this study was to determine if the X supplement increased muscle mass versus a placebo, is that applied or basic? Who thinks applied? Yeah, applied. So yeah, does this work to do this intended purpose? What if the question is, I want to take the ingredients in this supplement, let's say it's creatine, and use an animal or cell model to figure out which pathways the creatine interacts with to make muscles grow. that would be the basic kind of research that took you to get to creatine. You don't always have to do both. Like, so I'll say like sports performance is usually one step ahead of the actual basic research. So we've known for, you know, decades and decades that if you drink a bunch of chocolate milk and lift weights, you're probably going to gain muscle mass. What's in chocolate milk that does that? It got protein, you got sugar, you know, like now we know like there's better ways to do it. But so I guess, you know, usually the science is a little bit behind, but then the science fine tunes it and you get more, even better applied research. So, all right, nice. I'm going to, every once in a while, I'm going to jump back like this so we can at least remember that we're all in class and I'm not just talking to myself, which I do when I record videos. That's also kind of funny. All right, let's go back to the shared screen. Okay, so we're back on my screen. So again, the lab that, you know, we run here does both basic and applied research. So keep that in mind. I'm going to kind of cover some of the things we've done recently. So some methodological advancements, kind of going back to that. We've done a lot of research on muscle fiber typing, and we'll get into this. If you're unfamiliar with fiber typing, remember you got fast twitch fibers and slow twitch fibers. You actually have all these intermediary type fibers too. The way we differentiate fast and slow is one way, that would be the contractile speed, but the thing that really dictates that's called the myosin heavy chain, MHC, or you might see it as MYHC, and that's a protein on the myosin that tells it to be fast or slow. So this is Dr. Andy Galpin. I want to bring him up a lot too. He was another classmate and colleague. He's down at Cal State Fullerton. He does some really cool, his 5-minute, 55-minute physiology videos on YouTube. If you haven't seen it, just Google Dr. Andy Galpin. He's got some good stuff. But we did some things where we were looking again at fiber types. This is from a paper we did where we use this technique called SDS Page. So don't have to worry about what it does, but basically a gel. So imagine like a porous gel that's like, if you've ever done in biology class like Western blotting or anything like that, it's really similar technique. What we do is we load proteins at the top in these little wells, and then we run a current through this gel, and it pulls the proteins through the gel. So imagine the gel is kind of like a sponge, and you're trying to run a current to pull something through the sponge. The big things will get stuck at the top. The small things will go farther down. So whatever goes farther down has a smaller molecular weight. So we know that type 1 fibers actually have a smaller molecular weight. There's one there. A 2A fiber, which would be your fast twitch, that's a bigger molecular weight. And then we even have a 2X fiber, which is the biggest molecular weight in humans, and that's a super fast fiber. And then we've got these intermediary types or intermediate types or hybrid types. All people say all these different things. I usually call them hybrids, but they're just a mix between two different fiber types. All right, again, then we got slow to fast. And our labs published some papers, things like looking at how we can improve efficiency of this, because you can imagine how tedious it is to isolate each individual fiber, put it in a well, run this down, and then say we have to do 100 fibers per person, per sample. This can become your life. It became my life for at least a year when I was in Indiana. I'd wake up in the morning and be super excited to go load up fibers and all day go to class waiting for my experiment to be done, only to come back at 9:30 that it didn't work for some reason, and then have to wake up in the morning and redo it. But we'll talk a lot about methods, because methods can make your research really efficient or not so efficient. So that's one example of that. Another thing that we do is we do muscle fiber imaging or cellular imaging in the lab. And we have a really cool confocal microscope in biology at SF State, which I would normally walk you all down to like next week and we get to hang out and play with it. But instead, I'll show you a video and we'll talk about it. But we do this microscope research, and we published this paper on this, basically an assignment that you could do in a lab class on how to study muscle physiology there. So those are just some examples of methods advancements that we've done. As far as applied performance, one of the coolest studies, I'm going to talk about this a lot next week, so stay tuned for that. And if you want to read the paper, it is I'll put a list of all the papers that I have too up on iLearn, but you can Google this real quick. We did a study on these monozygous twins. So if you remember, monozygotic twins come from one egg, dizygotic twins, two eggs. So monozygous are the identical twins. We've ended up finding, so I won't give you the whole backstory because I'm going to do that more next week, but we ended up finding a twin set of males that were in their mid-50s that had basically diverged lifestyles at age 18. So imagine they were born, hung out together the whole life till they were 18, And then it's not like they like didn't talk to each other, but one of them moved to the Midwest, one of them moved to California, one of them became a truck driver, like long hauls and stuff, and one of them became a track coach. So the truck driver had years and years of sedentary behavior, didn't really exercise much, and the track coach was coaching and also competing and doing Ironmans and all kinds of stuff. So we had this perfect, like, identical twin setup where they were just separated at adulthood, and then at 55 we came back and we tested, basically everything about them to see what was different. And we're still following up on them. Actually, our 5-year follow-up's coming up soon, so that might be, thinking about thesis projects, this could be something we could add on to. Also, I have this picture, I didn't even say, but this is Katie Bathgate. She was the lead author on this paper that we did. What do we find? So basically we found after 30 years of endurance exercise training that this guy did, compared to his twin brother, it was about 9% less body fat, lower cholesterol, lower glucose, a 30% increase in VO2 max, so 30% more aerobic fitness. And this is what really caught us off guard. We ran this like 5 or 6 times, those gels, 55% more slow twitch fibers, 55% more in the endurance trained twin. Now, I imagine we would get like 5 or 10% more. How much can you really shift your fibers? Well, imagine if you're 18 years old and you go and you start running distance for the rest of your life, versus if you're 18 years old and you go and start powerlifting for the rest of your life. Maybe you could make actually a 55% shift one way or the other. Or these two individuals were just genetic freaks and they were just super adaptable. So that's the other question that we're kind of looking at too. But anyway, this was really unprecedented. Here's the graph to show that, but you just see the percent fiber type distribution of those type 1 fibers. The trained twin was almost 95% type 1 fibers in his quads, in his vastus lateralis. So anyway, there's a lot more to talk about. I'll get into that more next week. So another applied thing that we've done research on is tapering in sports to see how well tapering works. So if you don't know, tapering is basically reduced volume training before you get to a competition. So this works in everything from powerlifting. You might reduce the volume before you start competing, maybe a couple days or weeks or however it works. And then in endurance running, even 3 weeks or 4 weeks out, you'd start decreasing your volume while maintaining the intensity usually. But we can talk more about that. But again, the whole idea is instead of just training and competing, you'll train, you'll decrease volume, and hopefully get some kind of a super compensation before you compete. That's been the idea. So that's the applied part. There's been a lot of studies that have done this. So then we went kind of and tried to figure out, well, why from the muscle level do you get better, faster, stronger from doing this? So again, we wanted to look at these muscle cell or myocellular characteristics and gene expression in these cross-country runners. We found a group of cross-country runners from Taylor University, a D3 school out in Indiana, but these individuals were pretty trained. What we did was we took a bunch of muscle biopsies and we did a single muscle fiber function technique. So this was one of my mentors out at Ball State, Kiki Minchev. He's from Bulgaria. This guy has probably isolated more human fibers than anybody on the planet, or he's in the top 5 because he's been working at Ball State since like 1990 and nonstop from like 9 a.m. to like 6 p.m. He's in there isolating fibers. His eyes are like going bad. He's like, it's crazy. I talked to him the other day and he's like, I can't do it anymore. I'm like, dude, you're like 60 years old. You need to quit this job. But anyway, what did he do? He was basically would tie a muscle fiber. So this is 2 millimeters, not centimeters, millimeters. That's how long the muscle fiber is. Tie it to a force transducer in a motor arm with a little tie. This is a suture. So if you've ever had stitches, you know, the stitches you get are actually a intertwined kind of material. It's kind of like circular and there may be 2 or 3. This is one piece of that twine from a suture that we would tie a muscle fiber on here. Put it in calcium and ATP solution. So if you remember, we'll talk about the sliding filament theory later, but remember you need ATP, obviously your energy currency, and then you need calcium to release basically the calcium to get in there from the sarcoplasmic reticulum to get into the area where the muscles, myosin and actin are, and then it just contracts. So that's what we would bathe it in. And then from there, we would look at things like the size, pretty easy. You can just visualize and measure the strength, the speed, and the power. So we can contract it at different amounts of calcium or different temperatures and things like that, and look at how fast and powerful it is. So the graphs that you'll get from something like this will look like this. So this shows power on the y-axis and force on the x. So if you remember, it's kind of like a force velocity curve, but we call this a force power curve. So here at 20% of your force, you'll have almost maximum power of your myosin heavy chain 1 fibers. And then here is your myosin heavy chain 2A, which is your fast twitch fibers. They're like 6 to 10 times more powerful. This just shows the power increase before versus after taper of those fast twitch fibers. So what we found in this group of runners was that there's a 3% increase in race performance. That's good. That's what you want with tapering. No change in VO2, so they didn't lose fitness. But their fast twitch fibers were 9% more powerful and 15% bigger after 3 weeks. So if you've ever tried to get big, try to get your muscles 15% bigger in 3 weeks. I don't think there's any way to do it except for to overtrain yourself and then taper. So that's like kind of what we found, that the type 2 fibers are super adaptable to training, overtraining, and detraining. All right, so you're getting the applied part, right? Tapering works. Now we're like, why does it work? Well, it seems like the type 2 fibers get huge. So why do they get huge? All right, now we're in a real basic question. So we ended up looking at the gene expression. So again, we're going to get into this when we talk more about biochemistry and stuff, but the genes that you express are going to dictate what proteins are created and stuff. Has anybody heard of myostatin? Anybody? Okay, so some people have heard of it. Myostatin is a regulator of muscle mass. It's a negative regulator. So the more myostatin you have, the smaller the muscle mass. The smaller the myostatin, the bigger the muscle mass. So it's opposite. So in this case, we found out that after taper, myostatin decreased significantly in those type 2 fibers. So like physiology, after the fact, it all makes sense. This doesn't always happen. It kind of happened to make sense this way for this study. So we showed that those myosin heavy chain 2A, the fast twitch fibers, showed favorable gene expression. Now, if you Google myostatin, you're probably going to come across something like this. Pictures of one of these bull whippets or one of the Belgian blue cows. These are actual animals that are inhibited with have myostatin inhibition naturally. Well, they're genetically engineered that way or bred that way. And this is a human. There are 2 known humans on Earth, and they're in their early 20s now. This picture was taken in the 90s of a neonatal 7-month-old baby with quads and with gastrocs visible. So this baby has a natural myostatin inhibition. Now, this is probably not good, or else more humans would have it. It's a really rare genetic mutation, and it might affect other things like cardiac muscle and stuff. So, thinking about this could be your superpower if you wanted to do one thing, inhibit myostatin. Well, only in like the muscles you want, probably not necessarily all of your muscles, but it does happen naturally. This might be where we got the idea of like, myths like Hercules and super strongmen and stuff like that. Maybe there were people in ancient history that had myostatin inhibition that were just jacked and they didn't even have to lift or anything. So myostatin is a hot topic. It's been hot for like the last few years, but keep an eye. I think there's going to be more research coming out on these two babies that they've been following over time. So now they're probably 25 now. They're probably 23 or 25. All right. Everybody good? We got a few more slides and then we'll take a break. Cool. So the theme for this fall is the muscle memory hypothesis. I'm going to do a little introduction today, and starting next week, we're going to get some weekly readings on this topic. So what is the muscle memory hypothesis? So muscle memory is not really a official scientific term. I'm going to start you off with all this stuff. Like if you were to go to like a muscle biologist and be like, oh, muscle memory, they're like, well, your muscle doesn't have a brain. That's dumb. So that's not what we're not talking about that. There's no universal definition for this. I'm going to give you our definition. And it's kind of like a colloquial term or like a term that people just like kind of a slang describing the process of learning and retaining a motor skill. So this is often used in motor learning, or motor skill acquisition. So you can think of I haven't ridden a bike in probably like a year or something. Like I don't have a bike in San Francisco, but if I go to my parents, I do. And I have no problem jumping on a bike and riding it. That's an example of motor learning, muscle memory, right? That's not necessarily what we're talking about in this class, but keep that in mind. That is a motor learning thing. So what we're talking about in muscle physiology is a cellular memory, kind of a cellular memory mechanism, I guess, residing in the muscle cell. So it's inside the cell and it's related to regulating muscle mass. So what we're talking about is can your muscles remember how to get big or how to get small? And like, I'll kind of get into the next few slides, but that's basically it. So that's a way that the muscles regulate themselves, or they remember how to go back to a certain spot. And this might be related to what I mentioned earlier, the muscle nuclei, also called the myonuclei, or the cell's control center. And I say maybe related because there's a lot of, we're going to get really deep into some conflicting evidence on both cases, whether or not myonuclei are really important or maybe not as important as people thought. So keep that in mind. And this is all real-time stuff. There are nerd battles happening right now in journals that I'm going to share with you, of editorials that are scathing, going back and forth, like the respected Dr. So-and-so did not bring up our other studies. Like, it's going to be really fun. But anyway, the idea is maybe the myonuclei are related to this memory mechanism. So myonuclei, again, this is a picture of one of my muscle fibers. I'll show you a lot of mine with a lot of nuclei, the blue dots on here. You can see zoomed in, they're usually like cylindrical, or kind of either the shape of a football or even a little bit longer and stretched out. Their shape is actually important too. We might talk about that later, but inside each nucleus, you have a full copy of your genome, which is crazy. So you take one nucleus, you have the genes in there to make a brain cell. You have the genes in there to make a skin cell. You've got like all your genes, but your muscles only use a small percentage of those genes. So keep that in mind. If you were to unravel the DNA in one nucleus, it'd be about 1 meter tall. And you've got 1,000 of those per 500 microns of muscle fiber. So if you took all of the DNA out in your whole body, you'd be able to go to the moon and back like multiple times, and if it was stretched out straight, because it's really, really tight in there. But the whole purpose of keeping them in there is, you keep all of your DNA and chromosomes, and they're all packed in there so they can be used or not used. This is where DNA can be converted into RNA, and then the RNA is the message that can be sent out to the rest of the cell. And then this is also where the subcomponents of ribosomes are made. So anybody remember what ribosomes do? You can shout it out if you remember.
They make the proteins, right? Like, yeah.
Yeah, that's exactly it. So inside your muscles, you got these protein factories. The ribosomes make the proteins. I was going back and forth. I wanted to talk nuclei or ribosomes this semester. We will talk about ribosomes because it's all connected, but our focus is going to be on the nuclei. But the nuclei is where ribosome subunits are made and then sent outside to build proteins. So nuclei are super important. There's lots of them in your muscle, so your muscle can adapt and repair, and that's why your muscles are cool. It's like, if you like Marvel comics, it's like the real life, Wolverine. You can actually regenerate and repair your muscles with these nuclei. All right, so an important thing related to this this hypothesis, this memory hypothesis, is myonuclear domain size. So let's pretend you're looking at the muscle fiber like head on. So you're looking at it straight on like that. It's a cylinder. It's not a perfect circle, but it'll be close. And let's say you've got 4 nuclei around the outside. So the myonuclear domain, sometimes pronounced or abbreviated MND, is the sarcoplasmic volume, or the cell's volume per nucleus. So in this case, let's just say that this whole diameter was or the area was 100 microns or something, 100 square microns. Each one of these would have a 25 square micron domain. So they're nice, evenly distributed. They're not perfect like this, but they spread out. It's kind of like if you've been to the desert and seen all the sagebrush or cactuses everywhere, they're like almost exactly evenly spread out because there's only so much water in the desert and everything in between died. That's kind of what I think about as a nucleus. You only have one in a certain area because you don't need that many. So they're usually spaced out pretty evenly. Well, what happens if you increase size? So you do a hypertrophy training program. Let's say you're untrained because of quarantine and COVID, gyms open up, boom, you're in there like 4 or 5 days a week, you get hypertrophy. The first thing that happens probably is the cell just starts to get big. But then what people think is eventually the cell will get so big that it can't sustain itself. So you need to add those new nuclei to kind of make that domain size back to its normal size. So this is like the crux of this theory. And I'll tell you, up until 2015, I was like 100% bought in that this happens all the time, no matter what. And now I'm not so sure. So telling you kind of what's still known as like the main hypothesis or theory. That's another thing. Hypothesis is just an idea. which is kind of like this right now. A theory means it has a ton of data to back it up. So a theory is not just another hypothesis. A theory would be like, so you've got like the theory of gravity, and you've got the law of gravity is the mathematical equation, but the theory of gravity is how gravity actually works with relativity and time and everything. Like, so we've got, this is still considered, I think, the, muscle memory hypothesis or muscle memory. It's kind of close to a theory, but not quite there. All right, so muscle memory with strength training. So another word we need to know is plasticity. It sounds like plastic, so it's really adaptable. Muscle is really adaptable with environmental changes. So whether you use your muscle or you don't use your muscle, it's going to change one way or the other pretty quickly compared to other cells. So here we got a graph with muscle strength on the y-axis. We could also replace this with hypertrophy. So keep in mind, strength and hypertrophy, so strength and growth usually correlate. If you get bigger, you're probably going to get stronger, but there's not 100% correlation, because think about other things that can be related to strength. Like you got neurological adaptations, you got tendons, you got all kinds of stuff, biomechanics, you got efficiency, like there's just everything that goes into it. But in general, let's just say these go up over time. All right, we've got a trained athlete and an untrained individual. As you'd imagine, they're probably bigger and stronger. Let's say the athlete detrains. So something bad happened. We all had to go locked down for the pandemic for months, lost all their muscle mass and hypertrophy. Now they're at the level of an untrained person. But let's say, all right, we're able to go back to the gym, boom, after a few weeks or a month or so, they're back up to where they were, no problem. Whereas if you were to look at the same model with an untrained person and they just went back to the gym, it might take them a long time to get to where that trained athlete was, and they might not even get there. Now you might be thinking, well, what about the, genetics of these people? Let's just pretend like these are actually like the same person. We just have a twin version of them. We have a trained twin and an untrained twin, or let's pretend like they're animal clones. Either way, if you're already trained, you're going to be able to bounce back quicker than if you're untrained forever. So that's kind of the idea of this muscle memory, is really a retrainability of the muscles to get bigger and stronger to where they were before. So a lot of these long-lasting training effects have been attributed to the CNS, to your, like we were talking about riding a bike. Sure, I can ride a bike, but can I ride a bike at 400 watts for, I don't know how many, 5 minutes? No, there's no way, because I'm not trained. But if I, train my muscle to that point, that's kind of what we're talking about, is more hypertrophy and strength. So this is the first paper that I came across. There are a few earlier ones, was called Brusgaard 2010. And now I'm going to go through the mechanism that he attributed this to. And that's where we're getting into our muscle memory hypothesis. So this is a classic hypertrophy atrophy model, kind of what everybody thought. I don't know why my colors change on here, by the way. Usually I do like a red fiber and blue dots, but I think my PowerPoint had some kind of weird setting. So we have a blue muscle fiber with orange dots. Let's say we get a stem cell. So the stem cell is really a satellite cell, which we'll get into in the next couple of weeks as well. It's going to fuse to the muscle fiber, and you're going to add new nuclei. Cool. We know we can do this. You do this a lot when you're a baby and you're developing, but even adults can do this. We can add new nuclei. And then let's say the muscles get smaller. The nuclei undergo what's called apoptosis, or cell death. The nuclei die off. And we get myonuclear loss. And then let's say we want to go back to hypertrophy. If we need to go back to the gym again, then the idea is you got to get more stem cells to fuse more nuclei to do that. And it goes around. This is what people thought for a long time. And this still might happen in a lot of examples. It might just be a matter of time. Maybe you'll lose nuclei in 3 months, maybe 3 years, or who knows. So this is a question. Does this happen? The other question is, this is what Brusgaard brought up in 2010, which I'll share this paper with you all. This will be one of the first ones we read related to this topic. We still have our stem cell fusion. We add more nuclei during our first training program. Then COVID hits, we have detraining atrophy. But their idea is that you don't lose as many nuclei as you thought. So when you retrain, instead of spending all this time and energy putting new nuclei into this system, you basically just recycle the ones you had before. So your muscles get smaller, in other words, but the myonuclei stay there. So that when you're able to retrain They're already there. You don't need to make any more. And then those nuclei can make ribosomes and they can send out signals and everything. And then if you want to get even bigger, of course, you've got to go to the next level, probably add more nuclei. And then this cycle can progress until physics has its limits and your muscles can't get any bigger just because you can't send signals that fast. So there is going to be a limit for everybody. You've probably seen them. If you watch like some of these bodybuilding contests, you're seeing like the limits of human muscle hypertrophy just because you only have so many fibers. And we'll talk about the idea of hyperplasia, adding new fibers later too. But anyway, this is the idea that's been going around for about the past decade. But there are some questions, and I'll get into a list of even more. Does this occur in humans? How long does this last? How is this mechanism affected by aging? Like, so some of these questions we had back then, and now I've kind of broken this up again into 5 questions. This class has a lot of 5-part things, so keep that in mind. So this fall, we're going to try to answer these big questions related to the muscle memory hypothesis. One is, does it occur in humans? Most of the studies are animal studies. Is it fiber type specific? So meaning is maybe the fast twitch fibers or slow twitch fibers are affected differently. How long could these benefits last? If you do add nuclei, is it stick around for 6 months, a year? This could be really important for things like WADA, the World Anti-Doping Agency, because we know that testosterone increases nuclei count too. So maybe you've been off of your anabolics for 2-3 years, but you're still reaping the benefits. So that's kind of another applied part of this. So how long does this last? How is this affected by aging? We know aging affects everything. Maybe this happens in younger people, but as you get older, it gets tougher and tougher because you lose some of the satellite cells. And then 5, this is the other big question. I think people have been focused on nuclei a lot because they're measurable. We can see them. They're pretty big. And we know they're important for lots of stuff. But are there other mechanisms that could explain muscle memory? All right. So these 5 questions do not have clear answers. And that's why you're in grad school, to try to figure them out. So, I'll tell you straight off the bat that in this class, I've had 6 publications come out of the literature reviews, and or the lit reviews have gone into other publications. So some of the stuff that you guys write in this class might end up becoming actually published, or it might become part of your thesis. So keep that in mind as we go through here. But these are the 5 questions I literally came up with this week because I was like, these are what we don't know still. So these are what we're going to try to answer. All right, break time. I think everybody needs a break. Let's